Some of the hot topics in optics and biology and medicine this year include the use of cellular phones for spectroscopy and microscopy, which is, I think, something everyone can relate to. There are a few interesting advances too in the way some authors have managed to manipulate aerosol droplets and done some optical manipulation on chips, which is in the direction of miniaturizing devices and having them sit on something the size of a cell phone or a handheld device, a portable device, and there are many interesting advances in that direction this year. It's already difficult to trap objects in solution, and trapping aerosol particles is somewhat more complicated because of the physics of the problem. This particular talk by Professor Horstman deals with the trapping of single aerosol particles inside of a miniature chip. That's a very interesting advance in the area, and I think that's going to be one of the trends to look for in the future. Miniaturized devices that do things that are at the moment very technically challenging. And I think new technologies are going to build on top of these current devices. I would keep an eye on that. Another great paper you don't want to miss is on electrokinetic traps. There's an exquisite control being demonstrated in this particular paper, and I would recommend you look into it. It's going to be a great demonstration of new techniques and manipulation of microscopic objects with a very high sensitivity and control. In the area of cancer research, we have an interesting device that can sort cells, for example, cells which are diseased from cells which are healthy, and the way that they can be discriminated is by looking at the light emitted from them or their fluorescence activity, and this device being presented is capable of separating those two cells automatically. It's a very small device with a very convenient footprint, and it can definitely be potentially used for discriminating cancer cells from healthy cells. One of the things that gets me very excited about FiO is that there are so many different areas of research in the same place that one actually gets to meet a lot of researchers who are in very diverse areas, and sometimes one comes across a surprising topics of research that actually overlap with your own area of research. So, for example, optical trapping is actually merging a little bit with optofluidics. There's a natural relationship between those two technologies that make it a very convenient thing to mix them both. And you can come across people working in optofluidics and people working on optical trapping, for example, and a lot of new ideas and research collaborations come out of these meetings where you meet with the leading researchers in the fields. Don't miss optics in biology and medicine.